Hi, and welcome to the LI-COR Biosciences webinar on Developing Targeted Therapeutics, What You Need to Know. My name is Dr. Gretchen Hubbard, and I am a Senior Solutions and Support Scientist, Cell-Based Assay Team Leader, and In Vivo Specialist for LI-COR. We have 4 panelists joining us today to help answer any of your questions during the webinar. Dr. Maheen Ferdas, Senior Solutions and Support Scientist, Dr. Carol Quayle, Solutions and Support Scientist and Field Marketing Consultant, Dr. Christy Ambrose, Senior Director of Science and Global Support for Biotechnology, and Jeff Harford, Senior Marketing Product Manager, Biotechnology. During today's webinar, we will discuss a number of topics, including what is a therapeutic, how therapeutics are discovered and developed, some of the preclinical research that goes into therapeutic discovery and development, what happens after preclinical research and in clinical trials. Then we will focus on preclinical research and some common therapeutic assays performed during this stage of research, some published research examples of those assays, and how LI-COR can help you in your therapeutic research and discovery. First, we will begin with what is a therapeutic and a few examples being used in the world today. A therapeutic is defined as a treatment, therapy, or drug. You've most likely heard of chemotherapy, which is a drug therapy frequently used to treat cancer. There are over 100 chemotherapy drugs on the market, all of which cause adverse side effects because they not only target cancer cells, but normal, healthy cells in the body as well. Therapeutics can be unlabeled or labeled with a fluorescent dye such as LI-COR's IR dye 800CW. These dyes can be used for visualization purposes in fluorescent-guided surgeries, or even as a photosensitizer for near-infrared activated photoimmunotherapy, which is the use of light to target and kill cells. Targeted therapeutics is a newer group of drugs in which only the target cells are affected by treatment, eliminating the effect of the therapy on normal cells and reducing side effects. There are several advantages of targeted therapeutics over traditional therapies. In addition to reduced side effects in the individual, these therapies can also have improved effectiveness. There are several targeted therapeutic examples that you may have heard of being used to treat human diseases. Erlotinib, also known as Tarceva, is being used to treat several types of solid tumor cancers, such as non-small cell lung cancer. It acts by inhibiting EGFR tyrosine kinase, which is involved in activating many cellular pathways such as cell proliferation. Pembrolizumab, also known as Keytruda, is also being used to treat several types of cancers, including Hodgkin's lymphoma and melanoma. This therapy targets the programmed death receptor 1 pathway and prevents cancer cells from hiding from the immune system and prevents them from proliferating uncontrollably. A third therapy is rituximab, also known as Rituxan. It's used to treat autoimmune diseases such as rheumatoid arthritis, as well as some types of cancer. In rheumatoid arthritis, this therapy targets CD20 on B cells and can decrease inflammation, which helps with the painful swelling associated with this disease. Targeted therapeutics are now being used to treat over 15 different types of cancer and other human diseases. Those are just a few examples of how targeted therapeutics are being used in the world today, and many researchers are working hard to develop more for various human diseases. Now let's discuss some of the process involved in therapeutic discovery and just how long this type of research may take and how much it may cost. The process of therapeutic discovery and development is a very time-consuming and costly process. It is estimated to take 10 to 15 years for one therapeutic and cost between $500 million to several $billion just to get one therapy to the market. Preclinical research involves a series of different stages, represented by the 3 stages on the left-hand side of the figure, which are focused at reducing the number of potential therapeutic leads from thousands to just a few. The promising leads identified by preclinical research will then be tested in human clinical trials. Hopefully, 1 or possibly 2 therapeutics will eventually be approved by the FDA and be available on the market. Let's go into a little more detail about the preclinical research involved in therapeutic discovery and development. Preclinical research begins with therapeutic discovery. There are several ways a researcher may identify a potential therapeutic. It could be new insights into a disease process that allow researchers to design a product to stop or reverse the effects of the disease. It could be from screening of molecular compounds to find the possible beneficial effects against any large number of diseases. It could be existing treatments that have unanticipated effects, or even new technologies, such as those that provide new ways to target medical products to specific sites within the body or to manipulate genetic material. During preclinical research, there are a lot of steps involved in which promising targets can be identified and developed to ultimately take promising leads into clinical trials. This involves discovery and screening, lead optimization, and preclinical studies, including both in vitro and in vivo studies. We will talk about these steps in more detail and focus on the research that takes place in preclinical studies. The first step of therapeutic discovery is to identify and validate potential targets which may provide a benefit to patients. A combinatorial approach is used in order to identify thousands of potential targets for further validation. Several target identification techniques exist. Genomics uses available tools to assign physiological functions to the wealth of genomic sequence information available today. DNA microarrays are commonly used to identify targets that are present or absent in a disease. Although this can provide some insight into target identification, a combination of other techniques is needed in order to identify the best potential therapeutics. Proteomics is the systematic identification and quantification of the complete complement of proteins of a biological system. This could represent cells, tissues, organs, or even an entire organism at a specific point in time. Common techniques involved are 2D SDS page and mass spectrometry. Bioinformatics is the collection, classification, storage, and analysis of biochemical and biological information using computers. Together, genomics, proteomics, and bioinformatic approaches combine to provide the best insight into potential targets which may serve as therapeutics for a disease. After potential targets have been identified, they must be validated through screening. Screening is essentially a sifting process to find the few targets in a large set that have a desired biological activity. Screening can include binding, functional, biochemical, and cellular or cytotoxicity assays. A main approach used is high-throughput screening, known as HTS, which will identify what we call hits, which have the desired effect in these specific assays. Confirmatory testing and evaluation of the initial identified hits is the beginning of what is known as the hit-to-lead stage of therapeutic discovery. Various assays can then be used to discriminate between identified hits and to determine characteristics such as dose response, binding affinity, and metabolic half-life. Hits with desired characteristics, including high affinity, selectivity, and efficacy, will then undergo limited optimization to identify promising leads. These promising leads then undergo what's known as lead optimization, which involves the synthesis of new leads, sometimes termed analogs. The goal is to improve potency, reduce off-target effects, and have leads with metabolic properties that are suggestive of reasonable in vivo pharmacokinetics. These promising leads will be tested in preclinical, in vitro, and in vivo studies to refine the number of candidates for human clinical testing. Before testing a lead in humans, researchers must determine whether it is toxic and has the potential to cause serious harm. Leads will be tested in cells in order to examine features such as drug affinity and selectivity. Cell disease models display some or all of the pathological processes that are observed in the actual human disease. Studying disease models aids understanding of how the disease develops and allows for testing potential treatment approaches. In vitro studies can help refine the potential lead candidates and give insight into which have the most therapeutic potential. Following in vitro studies, in vivo studies are performed to further evaluate lead candidates. These studies are essential to therapeutic development because they provide the ability to evaluate a lead's characteristics, including physiological and biochemical processes, such as adverse effects and drug-to-drug interactions that cannot be observed in vitro. There are a multitude of animal models of human disease states that can be used for these types of studies. Ultimately, in vivo preclinical studies will give you information about how a lead is absorbed, distributed, metabolized, and excreted from the body. This is often termed ADMET. Its potential benefits and mechanisms of action, the best dosage, the best way to deliver a drug, such as by mouth or injection, side effects or adverse events that can often be referred to as toxicity, how it interacts with other drugs and treatments, and its effectiveness as compared with similar drugs on the market. From the 5,000 to 10,000 initial targets identified during the discovery process to preclinical in vitro and in vivo studies, the number of targets is eventually reduced to just a few leads. These are often termed potential therapeutics at this point. These potential therapeutics can now be tested in human clinical trials, and hopefully their performance will be consistent with the preclinical data obtained. Now that we've discussed therapeutic discovery, lead optimization, and preclinical studies, let's briefly talk about clinical trials and the process involved. Clinical trials are defined as any research study that prospectively assigns human participants or groups of humans to one or more health-related interventions to evaluate the effects on health outcomes. The goal of clinical trials are to determine how a therapeutic works, how it's absorbed and excreted from the body, does it have the desired effect, and is it safe and non-toxic. According to clinicaltrials.gov, there are over 312,000 studies currently occurring worldwide. That includes both interventional and observational studies. About 14% of the therapeutics are estimated to successfully gain FDA approval after clinical trials. The potential therapeutics from preclinical studies can potentially undergo several phases of clinical trials. Phase 1 is a short study of just a few months, involving only a few patients, and it's usually used to determine whether the therapeutic or treatment is safe. If a therapeutic passes phase 1, it will enter phase 2, and that study can last from months to a few years and involve more patients. It's used to identify whether the therapeutic has the desired effect in patients. If a therapeutic passes phase 2, it will enter phase 3, and this phase can last for years to decades and involve thousands of patients. The goal of phase 3 is to determine if the therapeutic is better than what is currently available on the market to treat the disease. After phase 3, the FDA will review the drug and determine whether it is safe and better than current available treatments. If so, then it can be FDA approved and released into the market. Phase 4 clinical trials will continue to be ongoing even after FDA approval, and they're used to determine the long-term safety, effectiveness, and any other benefits that the therapeutic may have. The ultimate goal is to get a potential therapeutic FDA approved and on the market for patient use. The upstream processes involved in therapeutic discovery, lead optimization, and preclinical studies are essential to identifying the best potential therapeutics and treating human disease. Because of the importance of these preclinical stages, it is imperative that researchers have reliable and consistent assays that give accurate and dependable data. Let's talk about some of the therapeutic assays that are commonly performed and relied upon during therapeutic discovery and preclinical development. In vitro assays can be used to study cell signaling pathways, specificity, dose response, and even high-content protein profiling, which can lead to biomarker discovery. In vivo and ex vivo assays are also used to examine characteristics such as therapeutic uptake and clearance, biodistribution, and specificity. Now let's look at specific in vitro and in vivo examples of assays performed during preclinical research. Two in vitro assays used for preclinical research include near-infrared fluorescence Western blots and cell-based assays, including the in-cell and on-cell Western. For those not familiar with cell-based assays, they are plate-based assays in which protein targets can be detected inside the cell or on the cell surface in their cellular context. These assays detect whole well fluorescence and offer a higher throughput assay compared to the standard Western blot. Now let's talk about some specific examples of where these assays have been used for preclinical research. One example of a preclinical assay commonly used for developing therapeutics is knockdown or knockout assays. Knockdown or knockout of target genes can be used to facilitate the identification and validation of therapeutic targets and to elucidate biochemical and cell signaling pathways. RNAi interference, known as RNAi, CRISPR-Cas9, and TALENs are powerful tools used to effectively regulate gene expression and are critical in therapeutic discovery and development. In this illustration, under normal conditions, the ligand in blue will bind its cell surface receptor, which results in phosphorylation of the gray protein. Phosphorylation is indicated by green signal in the in-cell Western or Western blot panels. Red represents the signal of a housekeeping protein. On the right-hand side of the figure, Knockdown is used to decrease expression of the ligand, so no blue circle is present. With no ligand produced, phosphorylation of the protein in gray is decreased, and we see a decrease in green signal. Expression of the housekeeping protein is unaffected, which is indicated by a stable red signal. In this published example, the near-infrared Western blot assay was used to measure knockdown of the target protein SMAD3 by CRISPR-Cas9 in 2 primary cell lines. Beta-actin was used for normalization, and the blots were imaged on the Odyssey CLX. SMAD3 knockdown was observed in lane 3 for both cell lines when Cas9 and the targeting guide RNA, gRNA, were both present. Phosphorylation assays are also commonly used in preclinical studies. Aberrant protein phosphorylation is implicated in the onset and the progression of human diseases such as cancer and neurodegenerative disorders. It is important to have assays that allow researchers to detect multiple proteins in a pathway. This diagram represents the binding of a ligand or therapeutic, represented by the blue circle, to a cell surface receptor. This results in an increase in target phosphorylation in the cell. Phosphorylation is indicated by the green signal in the in-cell Western and Western blot panels. Red signal represents total target protein and remains constant even with increasing amounts of ligand. In this published example, the in-cell Western assay was used to measure epidermal growth factor, EGF, receptor phosphorylation in A431 cells when stimulated with increasing concentrations of EGF. Phosphorylated EGFR was normalized to total EGFR and detected on the Odyssey CLX. With increasing concentrations of EGF, we see an increase in EGF receptor phosphorylation. Understanding the processes that lead to programmed cell death, known as apoptosis, is of utmost importance in the study of many diseases, including autoimmune diseases, neurological diseases, and cancer. Caspase-3 protein has been implicated as an effector caspase associated with the initiation of apoptosis. The diagram shows how treatment with anisomycin, a protein synthesis inhibitor, leads to an increase in apoptosis, indicated by increased expression of caspase-3. In this example, the in-cell Western assay is useful for quantifying apoptosis in response to drug treatment. HeLa cells were treated with increasing concentrations of anisomycin, which resulted in caspase-3 activation. This is indicated by an increase in integrated intensity on the graph. The experiment was performed in duplicate, indicated by the blue and purple bars. For therapeutic discovery and screening, it is essential to understand how a therapeutic binds its target in or on the cell. Competition assays allow for therapeutic and target interactions to be measured, as well as information related to the degree of affinity to be determined. In this illustration, a labeled therapeutic, indicated by the blue circle with green label, will bind a cell surface receptor, and signal is measured by either the in-cell Western or Western blot assay. Unlabeled therapeutic, indicated by the blue circle without a green label, is then added and competes for binding to the receptor. This results in a decrease in labeled therapeutic binding and decrease in signal, which can be indicated by a decrease in the green signal. The red signal is representative of the total receptor protein, whose signal would remain constant even in the presence of added unlabeled therapeutic. This example of the competition assay shows how the on-cell Western can be useful when examining therapeutic and target interactions. On the left-hand graph, IR dye labeled EGF was added to A431 cells, and EGFR phosphorylation was measured using an Odyssey imager. On the right, unlabeled EGF was then used to compete to binding to EGFR. Normalized signals were calculating using cell tag 700, which allows for normalization to cell number, and this value was plotted on the graph as % maximum normalized signal. With the addition of unlabeled EGF, you can see a decrease in signal intensity, indicating that unlabeled EGF is competing for binding to EGFR. Dose-response studies are useful in vitro assays for assessing therapeutic toxicity and efficacy in preclinical studies. In this illustration, a therapeutic, represented by the blue circle, is added in increasing concentrations. As more therapeutic binds to the cell surface receptor, a decrease in phosphorylation of the target protein is indicated by a decrease in green signal on the graph. This signal can be measured by the in-cell Western or Western blot assays. The red signal on the graph is representative of total target protein, and this signal remains constant even in the presence of added therapeutic. The IC50 can be calculated by how much therapeutic is required to reduce the biological response by 50% in cells. This example demonstrates how the in-cell Western assay performed on the Odyssey CLX can be useful to determine whether a protein target is involved in driving tumor growth. Two compounds, a type 1 and type 2 inhibitor, were used to inhibit the activity of PTK6 kinase, and the IC50 was calculated in a breast cancer cell line. Based on the IC50, inhibition of PTK6 kinase was determined to not be an oncogenic driver of breast cancer, and therefore would not be a good therapeutic target. In addition to the in vitro assays already discussed, high-content protein profiling can be useful to understand the signaling pathways involved in human disease. DigiWest is a contract research service offered by LI-COR, which allows researchers to quickly screen targets and potential therapeutic leads against over 50 predefined signaling pathways. DigiWest allows for proteins to be separated via SDS page by size. Proteins are then transferred to the membrane and biotinylated. Sample lanes are then cut into 96 strips, representing 96 distinct molecular weight fractions. Proteins are eluted from the membrane into 96 well plates. They are then immobilized on the surface of Luminex beads. A distinct colored bead set is added to each well. Beads from all the wells are then pulled to reconstruct the initial sample, where a defined color code correlates with each molecular weight of the immobilized proteins. A small aliquot of the bead pool is then incubated with Western blot primary antibodies before a PE-labeled secondary antibody is added for signal generation. The sample is detected on a Luminex instrument and a protein profile is generated. DigiWest high-content protein profiling can be used for both biomarker discovery and lead characterization. In the study on the left, DigiWest was used for analysis of mechanisms and biomarkers of platinum resistance in ovarian cancer patients. Protein signatures distinguished platinum-resistant versus sensitive patients and revealed promising biomarker candidates. In the study on the right, DigiWest was used to perform a mode of action study of compounds in comparison to a reference therapeutic. In addition to the in vitro studies just discussed, in vivo and ex vivo studies are critical to identifying potential therapeutics for human diseases. A potential therapeutic lead that is labeled, for example with a near-infrared fluorophore such as IR dye, can be injected into a tumor-bearing mouse. The animal can then be imaged in vivo on something like the Pearl Trilogy, and tissues and organs can be examined ex vivo on the Odyssey CLX. These in vivo and ex vivo studies allow for specificity, biodistribution, and other characteristics of the therapeutic to be determined. In vivo imaging using the Pearl Trilogy allows for therapeutic specificity and clearance to be determined using as an example orthotopic models indicated by the left and middle images. Ex vivo imaging on the Odyssey CLX can then be used to examine biodistribution within tissues such as the kidney as shown in the right image. In vivo and ex vivo imaging on the Pearl Trilogy can also be used to determine the best delivery method for a therapeutic and biodistribution of the labeled therapeutic in the body. Mice bearing either uterine or ovarian cancer tumors were injected either intraperitoneally, IP, or intravenously, IV, with near-infrared labeled siRNAs. The siRNAs were directed against AXL protein, which is involved in ovarian and uterine metastasis. The top panel shows in vivo imaging of mice, and the signal is strongest and most localized in mice-injected IP. Tumors and other organs were then excised, and more probe localization is indicated by higher signal in those mice injected IP compared to those injected IV. This is indicated in the lower panel. IP, therefore, is the preferred method of probe delivery for this therapeutic. We have now seen examples of in vitro, in vivo, and ex vivo assays that are frequently used during therapeutic discovery and preclinical research. Now we'll focus more on how LI-COR can help you in your therapeutic research. LI-COR offers a complete solution when it comes to your therapeutic development needs. Not only do we offer imaging systems to help you measure your preclinical assays, but we offer reagents, technical support, software, and other resources in order for you to be successful throughout the process. LI-COR offers 3 imaging systems which may be helpful for your research needs. The Odyssey FC can image Western blots, while the Odyssey CLX can image both Western blots, cell-based assays, and ex vivo tissues and organs. The Pearl Trilogy is optimal for your in vivo and ex vivo imaging needs. All of these instruments have a large dynamic range with which they can capture all of your information in a single acquisition. In addition, they use near-infrared fluorescence to give you the optimal sensitivity for your experiments. If you're not familiar with near-infrared fluorescence, there are several advantages that you can have by using this technology. Near-infrared fluorescence eliminates the variable signal obtained with enzymatic reactions that use HRP. In order to measure signal from the IR dye fluorophore, a solid-state laser is used for excitation, and the stable emission of signal can then be measured. Eliminating all of the variables associated with an enzymatic reaction, including time, temperature, and substrate concentration, leads to more consistency in your experiments. In addition to signal stability, using near-infrared fluorescence results in lower background from the membrane itself, often termed autofluorescence. This example shows a comparison to visible wavelengths. A lower background is important for assays such as Western blots. In addition to lower background of membranes, lower autofluorescence from cells and tissues is also evident, and this is important for cell-based assays and in vivo ex vivo imaging. Having a lower background plays an important role in obtaining the sensitivity that is desired for these types of assays, and this is why near-infrared wavelengths are preferred for both membrane and cell-based assays. Not only is the background dramatically lower using near-infrared fluorescence compared to visible fluorescence, but the signal-to-noise ratios of the fluorophores are also higher, and this enables high sensitivity for these assays. On the left, IR dye 800CW was conjugated to streptavidin, and it's being compared to Cy3 and Cy5 streptavidin conjugates. Cy3 and Cy5 are both visible fluorophores. IR dye 800CW fluorophore has a higher signal-to-noise ratio over a larger range than both Cy3 and Cy5. On the right-hand side, IR dye 800CW and IR dye 680RD, both LI-COR dyes, are shown to have high signal-to-noise ratio over very large ranges. This indicates that these IR dye fluorophores are optimal for both membrane and cell-based assays. For in vivo imaging, it's important to not only get deep penetration of laser light into tissues and organs, but be able to measure the emission signal from the fluorophore back out of the animal. The near-infrared wavelengths used for the Pearl Trilogy, around 700 and 800 nanometers, are where water, hemoglobin, and other tissue components have reduced light absorption. This allows for maximum penetration and sensitivity using near-infrared fluorescence. Several optical probes have been developed and optimized for both in vitro and in vivo preclinical research by LI-COR. In addition, you can label virtually anything with our dyes, whether it be an antibody, a peptide, a protein, a cell, a nanoparticle, and many others. Easy-to-use column-based conjugation kits are available, as well as click chemistry reagents to help you obtain the conjugated probe you want for your specific assays. In addition, several LI-COR dyes are in over 21 clinical trials. Their uses include fluorescent-guided surgery, photodynamic therapy, and ureter identification. For anyone doing preclinical or translational research, IR dyes can help you reach your research goals. LI-COR offers a complete solution for your research needs. We have over 18 years of near-infrared experience, as well as over 11,000 publications using our LI-COR instruments. We have the knowledge and support you need to help you reach your goals. We also offer a free education portal for individuals wanting to learn more about near-infrared fluorescence and applications. We provide free on-site training for instrument owners and users, and have online support including protocols and troubleshooting guides. We have two softwares to help you with your research needs, Image Studio and Imperius Studio. And these softwares can take you from image acquisition through statistical analysis to give you publication-ready data and figures. The therapeutic development process is long and tedious. There's a lot involved from discovery and screening through lead optimization and preclinical studies. To be successful, you want to have confidence in the assays you perform and the data you obtain. LI-COR has the tools to help you with all of your research, whether it's quantitative Western blots, cell-based assays, in vivo and ex vivo imaging, high-content protein profiling, and more. LI-COR offers the comprehensive solution from imagers, reagents, support, and software. In addition, we offer DigiWest Contract Research Service, which can help you quickly narrow down your potential therapeutic leads for your research, where time can be an important factor in obtaining funding and publishing your data. LI-COR is here to help you in your targeted therapeutic development, whatever your needs may be. If you would like more information regarding targeted therapeutics development, please fill out the request form on our website. If you have any specific questions, please feel free to contact me at gretchen.hubbard@licor.com. I'd like to thank you for attending our webinar today. As a reminder, anyone who registered will receive an email with a link to the webinar recording within 48 hours. Next, we will take a few minutes to see if there are any questions from our audience that we can answer. Okay, so the first question we have is, how do the IR dyes compare to others in the clinic, such as Psi-3 and Psi-5, and are they FDA approved? IR dyes have a high signal-to-noise ratio and are very photostable. None are currently FDA approved, but they are being used in over 21 clinical trials. It's important to remember that anything that is conjugated to a fluorophore, whether it's Psi-3, Psi-5, or specifically an IR dye, would have to go through the FDA approval process for use in human clinical trials and then onto the market. The next question we have is, why would you use DigiWest high-content protein profiling rather than something like RNA-seq? Well, high-throughput RNA sequencing, known as RNA-seq, does provide better quantitative measurements and more extensive gene coverage than proteomic-based assays such as DigiWest. In DigiWest, you're directly measuring the protein concentrations. But there's a lot of variations in transcript abundance that do not necessarily reflect changes in the corresponding protein abundance. So therefore, oftentimes both approaches are used to best correlate the data obtained at the RNA level with changes associated in protein levels. A third question's come in. I have a peptide that has shown promise in reducing proliferation of cancer cells in vitro. What options do I have to label this compound and track its biodistribution in animals? So we have several options at LI-COR that are available for conjugating a peptide for in vivo tracking. We have protein labeling kits that are column-based, and they allow you to label anything larger than 14 kilodaltons. We also have click chemistry reagents and free dye that you can purchase to conjugate yourself or pay a company to conjugate it for you.
I would recommend sending me some information about your peptide that you want to label, and our chemistry group can give you some suggestions about the feasibility and what method they would recommend for labeling. One question that's being asked is, can I just use a plate reader for cell-based assays rather than an instrument like the Odyssey CLX? Well, yes, a plate reader can be used to read a cell-based assay. However, traditional plate readers do not measure fluorescence from the entire well. Several spots within the well are measured and then the signal is averaged. The Odyssey CLX will measure the total well fluorescence, which is representative of the entire population of cells within the well. And this will really give a more accurate quantification of the protein amount in that cell population compared to just an average of several spots. At this time, we will begin to wrap up. Our panelists will remain online for the next several minutes to answer any of your questions you may have. So feel free to type your question into the chat panel. If you have any questions about the webinar, please feel free to reach out to me at gretchen.hubbard@LI-COR.com or to our biosales group at biosales@LI-COR.com. Thank you again for attending this webinar.